The second part of the exercise involves transferring patient cell with siRNA molecules in order to silence the expression of oncogenic EGFR and HER2 genes. Both EGFR and HER2 belong to the tyrosine kinase family and to the epidermal growth factor receptor family. Their hyperactivity causes increased DNA synthesis, proliferation, cell differentiation, inhibition of apoptosis, and consequently leads to increased metastasis and treatment resistance. In today's exercise, we will try to inhibit both genes at the same time because cancer cells have the ability to compensate for the lack of one growth factor by overproducing the other factor. Small interferating RNA molecules introduced into the tumor cells with a liposuction reagent called Hyperfect will be used to inhibit expression. Introducing siRNA molecules into cancer cells is not easy because siRNA molecules, like any nucleic acid, have a negative charge. There is also a negative charge on the surface of the cell membrane. Therefore, the transfer of native siRNA across the cell membrane is impossible. In order to counterbalance the negative charge of the siRNA molecules, the liposome was used in this case. The liposome has such a high positive charge that despite the balancing of the negative charge of the siRNA molecules, it remains slightly positive and can interact with the negatively charged cell membrane. Lipoplex formed from the fusion of siRNA and the liposome passed through the cell membrane by endocytosis or through membrane fusion. Cells prefer the endocytic route because in this case, unlike membrane fusion, there is no loss of cell membrane integrity. The liposome gets trapped in the endosome, but due to acid hydrolases, it is released and siRNA molecules are on the inside of the cell membrane. Inside the cell, siRNAs are recognized by a complex called RISC. This complex has two activities, helicase and nuclease, due to the Argonaute 2 protein. The helicase activity allows the discontact from each other, the strands, and Argonaute 2 degrades one of them. The strand that is an exact copy of the fragment of the mRNA molecule that we went to destroy is degraded. As siRNA molecules are double-stranded, there remains a complementary strand to the damaged one, which can join the mRNA on the basis of complementarity. The siRNA-mRNA complex is recognized by RISC and the process repeats itself, but now the mRNA itself is degraded. After the mRNA molecule is destroyed, the remaining complementary siRNA strands binds to the next mRNA molecule, and so on. Thus, we can see that one siRNA molecule can destroy many mRNA molecules. The lack of mRNA means the lack of the possibility of translation, so gene expression is inhibited in the post-transcriptional process. Before proceeding with the transfection itself, the transfection complexes must first be prepared. For this purpose, we prepare two Eppendorf tubes, one for the transfection complex and the other one for the control complex. The transfection complex consists of an anti-EGFR siRNA molecule and an anti-HER2 siRNA molecule, as well as the Hyperfect liquid reagent. The control complex, on the other hand, contains a control siRNA molecule and a lipid reagent. The control siRNA is not complementary to any mRNA in the cell, and the control complex itself is intended to check only the transfection procedure itself, whether it affects in any way cell viability or gene expression. We incubate the created complexes and then transfer them to flasks with cell cultures. Analysis of the inhibition efficiency of oncogen expression is then performed using flow cytometry and fluorescently labeled antibodies.